Okay. Hi, everybody. I'm Annalisa Zuccotti. I'm leading communication and events at BioRN, and I'm here today for M2M, a member-to-member interview. I'm here with Lucas and Fabian. Welcome to our office. Please shortly introduce yourself, what you're doing at Ariadne AI. Please.
So, hi, I'm Fabian Slava. I'm the CEO at Ariadne AI, and I'm also one of the original founders of the company. And today, I'll have an opportunity to basically talk about every aspect of the company, I think.
Thank you very much. You, Lucas?
Hi, thank you for having us here. My name is Lucas Schütz. I'm a biologist by training with focus on automated microscopy. And I joined Ariadne now 1.5 years ago. And as COO, I take care of the daily business that we do.
Okay, perfect. So if you have followed in the previous months the M2M format, so you know that we have an icebreaker question. So we have here on the table M&Ms, reminding us the name of our event, and gummy bear. So today is particularly difficult because you have to agree what you bring back.
So
you have to fight over chocolate or gummy bear. So what do you bring back to the office for your colleagues?
It would be easy if we had any vegans, but I don't think we do.
No, gelatin, no, and chocolate, no, sorry.
Personally, I would go for the gummy bears, but I don't know if that's something that Lucas can be.
Can you confirm on that, or you're going to fight?
I'm happy with the gummy bears. Also, I think M&Ms would be like a perfect match to our colored nuclei that we segment.
I see. Okay. Let's come back to the question at the end of the interview. Okay.
Controversy here.
And see what to choose, or I will choose that for you. Okay, so at the beginning, we normally ask some question about the history of Ariadne. I know we do not go back 30 years, but less than that. So if you, Fabian, you want to tell us, you have said already you are one of the founders, so where the idea came, by whom was funded it, what was your motivation a few years ago?
That's a great question. So, yes, as you say, we don't have to go back 30 years in the case of Ariadne, but we do have to go back a couple of years, about 10 at this point. And the company was originally founded then in 2014 by Adrian Manner, Jürgen Kornfeld, Emanuel Thaler, and myself. And three of us, so all of the names that I just said, except for Emanuel, who was our lawyer, were at the time working in a particular subfield of neuroscience called connectomics. And connectomics at the time was at a stage where rapid progress had already been made and was still being made on instrumentation. Instrumentation meaning that it was now possible to acquire very large volumetric electron microscopy data sets at very high resolution from neural tissue. And the promise of those data sets was that you would be able potentially to extract the detailed wiring, so the synaptic connectivity between neurons directly from a sample of neural tissue to understand how the circuits in that piece of neural tissue can achieve the types of computation that the brain is able to achieve. So, as I said, the instrumentation had come a long way, but Jürgen, Adrian, and myself were doing our PhDs at the time and still optimizing aspects of the raw data acquisition, and that turned out to be maybe the first half or so of our respective PhDs. But then what became more and more clear was that once you had the data, you just had the next problem, namely what to actually do with the data in order to extract meaningful biological insights from the data. So we had already spent quite a lot of time, many years, working on annotation software, so software specifically geared towards manually extracting wiring from volumetric EM data. But we also wanted to, but what that required, when I say manual annotation, what that required was a big team of annotators who would then go in, use that software, and actually reconstruct the structures in the data sets manually. So that's how Ariadne first started, that we built at Ariadne teams of annotators with that specific purpose of going in and manually reconstructing structures from volume electron microscopy data. But of course, that at the time that was necessary because automation just wasn't that great yet, and doing it manually was still the best way to do it. But of course, it didn't stay that way. It couldn't stay that way. And Ariadne also started investing more and more into automatic analysis techniques for volume electron microscopy data at first, so reconstructing organelles, synaptic structures, in volume electron microscopy data, but then also progressively more and more branching out towards other microscopy types as well. So our origin story is really very strongly about volume electron microscopy for neuroscience applications, but that is in some sense more of a distant past at this point. We've yet now branched out to more and more newer imaging modalities as well. We still do the manual annotator teams, but we also develop automation tools for all sorts of biomedical microscopy analysis challenges.
Yeah. So if I understood properly at the beginning, Ariadne was founded based on needs that you had as PhD students.
Exactly.
And this brings me to the next question, maybe for you, Luca. So what have been the major efforts and development that has been done in the recent years? So you have joined also more recently, but what from this idea, from 4 PhD students, how it developed?
Well, I think we underwent quite some development, especially last year. So I joined end of 21 with the same thought in mind that Fabian more or less presented. So I was looking for a new job, and when I saw Ariadne, I thought, okay, they're solving the problem that I had in my PhD. I come from a completely different field. I worked on developmental biology data with light sheet microscopes, where you also have huge data sets which you have to analyze.
Yeah.
And then I went more and more into automation. And when I was looking for a new job, I thought, okay, that's what I want to do at Ariadne. And then I thought, okay, I can bring my expertise in light microscopy in. And then last year, we started growing the company, especially then with new employees. Then also Fabian joined full-time as CEO in August last year. We got money from the Swiss government, which I think we received the confirmation beginning of 22. At the end of 22, we got money for another project from the state of Baden-Württemberg. And with all that new customers, we could really grow the company from having 5 people in the office to having like almost 15 people in the office. And we also went more diverse, more different data, and offering our services also to more people. Yeah.
Perfect. So what is the mission today of the company? So it was founded, we heard, developed. So what is the mission today?
Yeah, so in just a few words, it's about accelerating and automating biomedical imaging analysis for our clients. So anyone who's facing this challenge of spending too much time on data analysis versus actual data acquisition in the lab might potentially want to talk to us about, taking away that bottleneck. And so one thing I want to highlight is that we don't necessarily build off-the-shelf software. And there are many, there's good open source software out there, such as ImageJ, Fiji, which I'm sure almost anyone who's doing biomedical image analysis has heard of or has worked with already. And for those types of simple one-off analysis tasks where you can work with that software, that is often just good enough. But then there's a whole class of projects that are not feasible without fairly sophisticated automation, or even combinations of automation plus manual annotation. And that is exactly the service that we can offer, that for a new project, a new analysis challenge that is exactly in that category, we will build something custom-made that solves exactly that.
Okay,
Yeah.
perfect. So the field, obviously, of imaging analysis is very dynamic, and I think you reflected that also in your development from the foundation to now. So what has changed over the years? Maybe Lucas, you want to?
I think what has changed is that automation on the microscope side is way more accessible than it used to be. It used to be something for big imaging facilities or highly specialized labs. But this has totally changed. I worked in light microscopy quite a lot on automation on programming microscopes, and it's by now so easily accessible because it has been integrated into the microscope control software that everyone who can operate a microscope can also set up an automated experiment, which takes hundreds of gigabytes of data or even more. But nobody or only a few people think about what comes behind them.
Yeah.
So how do I analyze this data? Because that's not as easy. That's not set up yet. And this is where Ariadne is placed. This is our mission to solve this bottleneck.
Yeah, perfect.
And
And
can I add one thing actually to this, because I want to add one thought just. Also, AI technology specifically for image analysis has come an incredibly long way. And a lot of people know this if they work with this field more narrowly, but maybe sometimes there's a sense that there's a big AI hype and it's kind of overhyped. But in this particular field, there's, I would say, no hype. Like, we had a team of 100 people just for analyzing those electron microscopy data sets that I mentioned earlier. And to do the same work, you would now need less than 10 people, just because the AI gives you such a high leverage. Or, of course, the same people, the same 100 people could do the work of 1,000. And that's what's happened in 10 years. And so it's made a big difference.
Yeah, perfect. Thank you. And we will take now the time definitely to go into details and to explain exactly what you can offer. I wanted to remind to the people that are watching live that you can post your questions also in the chat. Just write them there and I will read them to Lucas and Fabian soon. So I promised that we would go into detail, but we always make this exercise. So we give first an appetizer about what is innovative and what distinguishes the company, your company, from others. And then we go into detail and we really look and we take time to understand the innovation behind your offer. So for you, Fabian, the appetizer, so if you would describe what is Ariadne and why it's different and from other companies, what is your USP? There.
So I think we have a very unique team. We have an AI team of biologists or a team of biologists who are AI experts. There's not many other companies or other teams like that, I think. And what that means is that we're able to think about biological challenges that our clients are trying to solve from their perspective as we develop the software for them. And that allows us to develop something that is custom tailored, not to, we're not trying to in any general fashion, solve a problem in the general case. We just want to understand what the client wants from us and then solve exactly that for them so that they can spend more time in the lab and less time at the computer.
Yeah, and since your team is at the same level as the scientist that needs you, probably it goes much faster than in other cases.
Exactly.
When you have only IT person or AI expert that are not biologists, for example, this would be much more difficult.
Exactly. From the perspective of our clients, it's simply what that means is that they are able to access a high-end AI team specializing on biomedical data that if they had to build it themselves would probably take years, and the project would take quite a lot longer than if we did it.
Yeah, perfect. And now for Lucas, you have the chance to go a bit more into details or give us some insights into the service for your customer. for your clients, the innovation behind these services?
So I think we'll continue a bit what Fabian said. It's about the team, and the one point which I want everyone to think about is, where does your image analysis start? It starts actually not at the computer once you have your data. It starts in the wet lab when you prepare your samples. And then it continues at a microscope where you set your settings. And then only later, once you have your images acquired and happy with them, you can analyze them and then do the data science on it. And it's a big part where, which is not always taken into account. And that's a problem later on with image analysis. And what we can provide really in our team is we cover the full expertise from the wet lab to the data science via all steps. So we can think about the whole experiment and not only a part. And we do that actually a lot, especially in initial customer calls. When we look at data with the customer, sometimes we consult on imaging, how you can take better data to be able to analyze in the end what you want.
Okay, so you go back even to the sample preparation.
We consult even on sample preparation and imaging and so on. And this is one part of our innovation. And the other part is, I would say, really our team who learned working together over the years, and they have different professions. So we have biologists, we have the developers, studied computer science, we have physicists. And I can really only tell from my own experience, I worked with a theoretical physicist as biologist at some point, and we took half a year until we could really understand each other when talking about the same thing. And this is what we have in the team today. We have an extremely diverse experience, but everyone understands each other when they're talking to each other. And that's why we can choose the right method for each customer. Because I mean, today it's very easy to do AI. You just like Google AI, and then you take the next best image analysis algorithm and do something with it. But that doesn't mean that you get a good result in the end. You get maybe 80% of what you want. To do the rest of that, you need to have really a great team which has experience, knows what it's doing, knows which algorithm it is, knows how the ground truth has to be done, and also knows the biological point of view, what is actually signal, what is an artifact, where do we have to be careful with the image. And all this we combine in Ariadne, and this is what makes us special.
Yeah, thank you. You are just, I'm having deja vus of my time as a scientist sitting under the microscope collecting data, and then people were telling us this microscope now can take the image like this and like that, and I was like, what do I do then with these huge images? How can I analyze? So but this goes more than 10 years ago. So I'm happy that now scientists can count on you also for that. So more technical questions. So Fabian, what types of image data, experimental image data, do you work with?
Yeah, so at this point, we're almost image type, microscopy type, preparation type agnostic, I would say. So for the first couple of years of the company, there was a focus on three-dimensional electron microscopy. We still have great tooling around three-dimensional electron microscopy, but it's by far not the main type of data set anymore. So we've accumulated considerable experience with three-dimensional fluorescence light microscopy, such as from confocal microscopes. Something that has become very relevant and a bit of a recent focus for the company is two-dimensional multiplex immunohistochemistry data. So those are in XY fairly large sections, ultramicrotome sections typically, where you have a large array of channels mapping out specific markers on top of that in the same coordinate system. And then you want tooling around registering the channels, detecting individual cells, segmenting them out with very high quality, and then mapping all of the various specific market channels onto the single cell level and doing downstream statistical analysis on this. This is something that's very relevant and very common data type to us now as well. We've also worked with data from the clinic, CT, MRI. That is at this point probably rather a minority of the data sets that we've worked with, but we have experience with that also. To summarize, I would say that the main focus, 80% maybe of the data that we work with, comes from preclinical research labs, either in academia or academic-like privately funded institutions, and industry. So preclinical microscopy data, I would say, is the main focus, but that is, of course, a big category that covers lots of different tissue types, preparation methods, types of microscopes that could be used for acquisition. A bit of a strong point, I would say, always, or maybe something that those data sets have in common, is that they tend to be large. Or small, but lots of them. So basically, I mean, that is probably in some sense what you would expect, because if it wasn't like that, you could do it yourself manually quickly. Yeah. But exactly in the situation where that is not possible anymore is, where our technology usually comes in very handy.
Yeah, perfect. So you mentioned already the size of the data set. Would you like to comment on that? So I know that it probably depends a lot on the so the aim, the objective of your support, but yeah, the smallest and the biggest range. Can you give us some inputs on that?
Yeah, sure. I think the smallest range of data we have is maybe around 1 megabyte, a bit less. And the biggest range is in the range of multiple terabytes for one data set. And it doesn't matter for us. We can really handle all of them. We also have the IT infrastructure to handle that. Actually, we just upgraded it to be able also to extend in a very large range, having storage up to petabyte, and also be able to handle multiple data sets in the terabyte range at the same time. And as Fabian already said, it's like usually 2 reasons customers come to us. Either they have too many files, too many small files, or they have a few individual big files. And it doesn't matter for us because our technology is designed in a way, for example, when viewing the data, you load only the region that you're currently looking at.
So which means they are big data.
Yeah, which means that even with a terabyte data set, you're only loading a few megabytes at the same time. And so we can really process anything. So it's not only for viewing, but also our processing algorithms are designed in a way that they can split the data into individual parts and then process this. So we are really not limited on that.
Okay, perfect. So if a scientist from one of the university or the research institution is coming with many small files, for example, confocal imaging or single cell, so how would it look like the process? So I get in touch with you, obviously it's saying what is the objective of this, so what do you need from the scientist to start the job?
Yeah, it's really, the answer to that is always the same. We would like to look at the data together. We find that this type of data usually speaks for itself in a way, just based on the experience that we've gained with diverse types of data that we've worked with in the past. We can usually fairly quickly, sort of eyeball what the likely analysis challenges are going to be. So that would be step 1, looking at the data, but looking at it together. Looking at it together means that we really want to understand what the question is, what the biological question is that the client is trying to answer using that data. Okay, so I mean, in an academic context, that could be a research question, but in an industrial context, that could be quantifying one specific endpoint in order to learn something about a certain drug that's in development, right? And that is very important to us, that we're able to understand research question behind the data, essentially as well as the client does, so that we can really take over the client's perspective while we do the development, that we can look at the data with the eyes of the clients while we optimize the end-to-end pipeline that we're building for them, while we optimize the algorithms. The thing is, if you don't do that, if you over optimize on a particular measure, such as let's say pixel-wise outline accuracy of the outline of some segmentation, but at the end of the day, what the research question requires an analysis of the number of cells that are present, then that would be effort very poorly spent. Rather, you should focus on false positives or false negatives on the object level without looking so much at the shape. That's just one, very trivial everyday example, but that is the kind of thing that we want to be able to do essentially at every stage of the development process.
Yeah.
I want to add something to that. I really want to encourage every client not to come when you have a lot of data image, but come early after you have a pilot experiment done with a few data, and then we can check your data, talk about it, and give you hints what would be ideal for really setting up the big experiment there, so that we can analyze it really as our clients want.
Yeah, perfect. So before you've talked a lot about the team and so on, we have a small video where we will see also the team members. But before that, there is still a question from the audience. So who are your customers? So can you give a proportion, industry, academia, and are academic entities typically able to pay for your services? And it's funny because we were talking about that just before connecting for the interview. Yeah.
So we have both. We have, and I think distribution is usually 50-50.
Okay.
So we have 50% industry customers, we have 50% academia. And we're happy really with both, because it's both interesting projects. Paying is on a different level. Of course, we really try to make a good price for academic customers, because we are still all scientists and we love the questions we get. So we really do an effort to make a good price there. But what we see from academic customers, that it's often difficult, because especially here in Germany, funds they have are often very restricted. So they might have 100K lying around, but it's for personal costs. So they have to hire a PhD student or a postdoc and cannot spend it on our service, for example. And just from my personal story, I remember that I was like, when I did my PhD, talking to my bosses and saying, yeah, you need to put in this grant like 10K, I need a computer to do image analysis. And so sometimes we also write grants with academic customers to get money specifically for analysis. Yeah,
perfect. And I think, this is changing a lot in the recent time, and everybody will need to hire, to subcontract companies to take over part of the project. Yeah. So now it's time of the promised video. So my colleague Stephanie was visiting you yesterday. She took the mobile phone and did some small videos, talked with your colleagues. So let's have a look.
Hello, I'm Stephanie from BioRN, and today I'm at ariadne.ai. It's a member of the BioRN network, and I would like to get some more information about the team and the solutions and the technologies. So I'm really very happy to meet today Fabian Swagner. He is CEO at ariadne.ai, and Lukas Schütz, the CEO at ariadne.ai.
Great to have you here today, Stephanie. Let's introduce you to the team.
Thank you very much.
Welcome. Let's come in.
Thank you.
Hi, welcome to ariadne.ai. At ariadne.ai, we automate and accelerate biomedical image analysis for our clients. So we'd like to introduce you to some of the team, some of the different roles that entails. And the first stop is going to be Matt, one of our developers who's working on a pipeline to analyze multiplex immunohistochemistry data.
In this pipeline, we are analyzing multiple marker channels showing different antigens with respect to different cell types. The analysis consists of 3 steps. The first one is registration, the second one is cell segmentation, and third one is custom analysis. Here, for example, in this example, you can see some of the functionality. You can define different types of masks to show regions of interest. You can align these different marker channels so that you can look at the regions where antigens match in certain patterns. And then you can also look at which cell types yielded which particular combination of antigens so that you can make some conclusions.
This is Tosin Oyewale, and he will explain in a few words what he is doing for us.
As he rightly described, I'm mostly involved in the segmentation aspect of the company. Colloquially, that is object identification. We have all kinds of data, including multiplex immunohistochemistry data, as my colleague previously mentioned. Here is such an example. In multiplex immunohistochemistry data, you have several layers, and you are supposed to identify objects based on either one or multiple layers. This means we have to identify the cells and generate what we call ground truth annotation. This is like an example that we provide for our deep learning pipeline to identify the object and extrapolate this to the entire data set. Yeah, for example.
All right, so last up is Sebastian, our web full-stack developer and visualization artist specializing on biomedical visualization. One great example of his work you can find here on the wall, journal cover that he designed. But maybe let's let Seba explain what he does in detail himself.
All right. My work is about visualizing our results, which usually means that we create 2D meshes of our image segmentations, and then we use these meshes to, for example, create images or animations for our customers, who can then use these renderings for their own research, or if they want to publish an article or something like that. And one nice thing about our image meshes is that they show the real data, how it was recorded by the microscope, and how our pipelines then analyzed it. So we don't do like a schematic rendering or something like that. We do not edit the meshes. They show the true sizes of these biological structures, and this helps researchers to, for example, visualize and analyze the relative sizes or like individual features of these organic structures.
So it was great to have you here. Thanks a lot. And for everyone, if you want to learn more about our services or about AI, come visit us in our office in Heidelberg or at the BioRN lounge. Or if you are from abroad, we go to multiple meetings. Next one is in Basel, Future Labs Live, and the Bio Convention in Boston. We look forward to seeing you.
Perfect. That was really nice to have also an impression. You talked a lot about the team. Now we have also some faces. Yeah, very nice. Thank you very much. So I think we talked a lot about your innovation, your services, how you do that, what is the specialties. But you are also, since 1 year almost, member of BioRN, of a cluster. For you, Lucas, the question, so what was your motivation to joining the cluster, and how can you help the other members? I think you covered already a bit, but yeah, maybe now strengthening this sentence. Yeah, so what can you offer to the other members of the cluster, and why you are member here?
So I think our motivation to join was really being part of this excellent network here in Heidelberg. We see in multiple sites that Heidelberg is really a good place for us to be, especially with hiring new people or contact to other companies. And there are also a lot of biotechs in Heidelberg that we know. And so this is one reason. And what we can offer really is we fill the gap that comes after wet lab automation. So if you do screening applications, anything like that, wet lab automation, and you have image data, we are the right people to talk to.
Perfect. And the other side, so what members our members can help you. So what are you looking for? So what do you see? Where do you see the potential?
Yeah, so I think there are a bunch of different ways in which the network could be beneficial to us in this direction. So one way is just, I mean, very simply learning about what people do, what people's requirements are, and how we can build tools to, you know, solve those challenges or make them go more smoothly. That's just, you know, learning about what other people do and what their challenges are. But then one thing that we're also always interested in is finding partners to co-develop solutions. So specifically, I mentioned that we are working on spatial biology, so the multiplex immunohistochemistry data set, and a little different from our previous modus operandi of building custom tailored solutions for specific problems. Here we're actually in the process of developing software as a service solutions. So basically something that will, a tool that will be accessible through a web browser that will allow clients to do this type of analysis on highly multiplex 2D data sets and down the line 3D as well themselves. So this tool is one that we're developing ourselves, but we see a lot of potential in accelerating it by finding partners for co-development. Yeah. That is maybe something that I would like to highlight. And then also specifically with an eye to accelerating the progress on this software as a service tool, talking to investors would also be helpful.
Yeah. Okay, perfect. Meanwhile, is another question from the audience. So I assume you need labeled ground truth data to fine-tune your neural networks on the customer's data. Does the customer provide this data, or is this part of your service?
Yeah, so this is a crucial and absolutely integral part of our service. So one thing that I haven't, or we haven't talked about very much today is that in addition to our custom tailored workflow development services, we also have the annotation team service. So basically we can rent out access to our annotator team to others, but importantly, we also have access to the team ourselves, of course, in order to very rapidly create exactly that high quality labeled ground truth for any type of data that our customers throw at us, basically. And this is really essential. So in this type of deep learning based approach to image analysis, there's not really, how should I say that? The idea that you just develop a better model and then suddenly the results get much better has some degree of merit, but it's just a small part of the story at the end of the day. What's at least as important is having access to large amounts of very high quality ground truth in order to train the models. This is going to make or break any algorithm that's based on deep learning, no matter how modern or fancy the model is. And then the third component, I already highlighted that, I just want to say that again, in that context is then you also need detailed biological understanding of what the question is in order to do the analysis the right way. But either way, the point being, high quality ground truth is absolutely essential for everything that we do. And if a client comes to us with a new type of data, we would ideally like to see a little bit of ground truth already, just as an example, for us to understand, you know, if the data is very different from something that we've worked with before, what does the client actually want to see in there? So in that sense, maybe minimal amounts of ground truth could be helpful, but it's actually optional. So we can also just, you know, do it in a call and the client points out a bunch of structures and then we learn about it in this fashion. It's also totally okay. But at that point, we would then generate a lot of ground truth ourselves. Yeah.
Okay, perfect. So I think our time is running up. And we still have the open question. So I have seen, actually, you were right, the colors on the screen of one of your colleagues. So you need to take this. So M&Ms are is the right choice for you and your colleagues. And yeah, for you, Lucas, a last question. So you can nominate the member that could be on our list to be M2M. So who would you like to see in an interview?
I think I would like to nominate Anavo Therapeutics.
Okay.
So Joe Lewis is working there, and he and I know each other still from Ample.
I see.
And he has also a very nice company. So yeah, I would like to hear more.
Thank you for the suggestion. We will take it on the list and connect to him. We know them also very well. Thank you. So before closing, so first of all, thank you very much for taking the time.
Well, thank you very much.
Yeah, thank you for replying to the questions, for your availability. And if you join our lounges, so last Tuesday in the month, every month at Urban Kitchen, you are going to meet one of them for sure, or one of your colleagues if you cannot join. And otherwise, yeah, I will send you the contact of them. You can contact them anytime to explore a bit how you can cooperate, how you can help them, how you can help the audience, and so on. And we see each other. So for the next M2M on June 21st, we will interview Cogitars. So we will stay in Heidelberg, probably going to their premises or inviting them here. Let's see how it goes. So thank you to the audience. Thank you to Ariadne AI, and see you in a couple of weeks. Bye-bye.
Thank you.
Bye-bye.
Bye.